Hello dear students, this is Dr. Devyani Sharma from the Faculty of Science, Sri Rawatpura Sarkar University, Raipur, Chhattisgarh. In this video lecture, we will be discussing about haptens and adjuvants. We will discuss about the similarities of antigens with haptens as vaccines and other topics related to immunology. So let us start today's lecture. So haptens, which are usually pronounced as haptene, it means to fasten. It is a Greek word and it is very low molecular weight. So this haptens can become very tightly fastened to a carrier molecule, which is mostly a protein, by a covalent bond. And this hapten carrier complex stimulates the production of antibodies, which the unbound hapten cannot do, and becomes immunogenic in nature. So the hapten then reacts specifically with the antibodies generated against it to produce an immune and allergic response. Thus, although the haptens cannot initiate an antibody response on its own, but it can bind with the antibodies and may act as antigen sometime. So, a very good example of haptens is penicillin, which is also a very small molecule and is not antigenic in nature, but as soon as it combines with certain serum proteins of sensitive individuals, the resulting molecule becomes immunogenic in nature and it activates the lymphocytes and also initiates a severe and sometimes fatal allergic reaction, which is a type of immune response. So the term hapten was first of all coined by Karl Landsteiner, and these haptens may be complex or simple in nature based on their valency. So the complex haptens are usually polyvalent in nature and they precipitate with specific antibodies, unlike the simple haptens which are univalent in nature and they non-precipitate with the specific antibodies. So in this picture we can see that whenever a rabbit is injected with hapten molecule alone, there are no antibodies raised. Rabbit, then the antiserum contains the antibodies raised against the carrier molecule. But the response here with the hapten carrier conjugate is little higher than the carrier molecule alone. So, this particular hapten, it initiates and elicits an immune response and is better in eliciting the immune response than the carrier molecule. So, three types of antibodies are generated whenever a hapten carrier conjugate is injected into a rabbit. There are antibodies against the hapten molecule, there are antibodies to the carrier molecule, along with there are antibodies to the conjugate of hapten and carrier molecule. So, this particular table summarizes with the immunization with the hapten carrier conjugate. So, whenever there is this hapten molecule injected, there are no antibodies formed. But when they are injected with a protein carrier or hapten carrier conjugate, then there are specific type of antibodies produced. So this was all about haptens. And there are certain examples of haptens which are very used nowadays. For example, aniline, which is an organic compound and its derivative of O, M and P benzoic acid. This was the first researched hapten. And there are other examples like urushiol, hydralazine, fluorescein, biotin, penicillin, digoxazines and 2,4-nitrophenols, which is another organic compound. So antigens and haptens are very similar in nature. They both are antigenic. They both are present on external cellular surface of the microbial pathogen and other agents. And they both antibody via weak linkages like the ionic interactions, hydrogen bonding and hydrophobic interactions. But the very basic difference between the antigen and the hapten is that the antigens are immunogenic in nature but haptens are not. So let us start with adjuvants. So these adjuvants are the substances which when mixed with the antigen and injected into a certain kind of host, it enhances the immunogenicity of that particular type of antigens. So this particular antigen and the adjuvant combination is used in the vaccine development process. So adjuvants were discovered in the 1920s by a French scientist named as Raymond. So Raymond noticed that the addition of certain substances, particularly aluminum salts, to the vaccines increases the efficacy of the vaccines. So since then, the adjuvants have become an increasingly invaluable component in the field of vaccinology. So the word adjuvant, which is derived from a Latin word adjuvare, it means to help. So these adjuvants often not particularly immunogenic in nature by themselves, but they serve to promote and enhance the immune response to vaccine components, thereby lessening the required dose of the said vaccine and prolonging the immunological memory of that particular kind of a vaccine. So adjuvants are really important in the vaccinology. So there are certain features which should be there in a particular ideal kind of adjuvant. For example, they should be safe, they should be stable, their bioavailability and the generation of long-term immunological memory is a must for any kind of an adjuvant to combine with the particular kind of a vaccine. And basically it helps in the activation of immune responses, may it be the T helper cell activation or B cell activation. It should be able to induce the cytotoxic T cell response also, and it should be able to promote the crosstalk between the innate and the acquired immune system. It should be able to prevent the autoimmune responses as well, and it should also induce non-pathological inflammatory responses. So these are all the features of an ideal adjuvant. So there are certain adjuvants which are very commonly used, as I just discussed in my previous slide, like the aluminum salts. So alum is the first aluminum salt which is used as an adjuvant, and now which is completely replaced by aluminum hydroxide and aluminum phosphate for the commercial vaccinations for better performance. And there are other adjuvants which are called as FIA and FCA. FIA stands for Fluent's Incomplete Adjuvant, which is oil in water emulsions, and there is another adjuvant which is commonly used, it's called as Fluent's Complete Adjuvant, which is inactivated and dried Mycobacterium tuberculosis. It also elicits and increases the immune response. There are certain adjuvants which are plant derived, like plant saponins, certain kind of cytokines, Quil A, which is a detergent, certain mineral oil and food based oil like peanut oil, squalene, which is a carbon organic compound obtained from the shark liver oil. So adjuvants act. These are like they extend the presence of antigen in the blood and helps the antigen presenting cell absorb the antigen in a better way. It also activates the macrophages and the lymphocytes, and it also supports the production of cytokines, which is very important for the generation of an immune response. So this was all about the adjuvant along with haptens. For any kind of query, please comment on the link here and put your questions there. Thank you for now.